AS Level Biology 9700, Paper 1, Variant 2, May June 2025. And this is the third video in which we will discuss the question number 21 to question number 30. So, question number 21 is the diagram shows the arrangement of chromosomes with attached spindle fibers during a stage of mitosis. Mitosis is a cell division in which chromosome number is maintained generation after generation. So here you can see the chromosomes which show correctly identify the stages of stage of mitosis and describe what is happening. And you can see the chromosomes are moving towards the opposite poles. They are get separated from each other because you can see in each case you can see two centromeres. Two centromeres shows they are get separated. The sister chromatids are chromosomes are get separated from each other. Now these chromosomes are moving towards the opposite poles. So this stage is called as anaphase, when sister chromosomes are separated from each other and move towards the opposite poles. So these can be the anaphase. So A and D can be the correct option. In metaphase, the chromosomes are arranged on the equator. There is one centromere, but here you can see two centromeres. So this is not the metaphase. So anaphase, anaphase, and the first B and C options are not correct. A, chromosomes are being pulled towards the centromere. This is wrong description. In the anaphase, the chromosomes are pulled towards the opposite poles. So A is also not correct option. Sister chromatids are being pulled towards the poles of the cell. Yes, they are moving towards the opposite poles. So this statement or description is correct. So D is the correct answer. Next question is, during which phases of the D cell cycle does DNA replication takes place? So DNA replication takes place. There are different stages, G1, G2, M and S stages. So during the S phase of the interphase, the DNA replication takes place. M is the mitosis stage and G1, G2 are the other stages during which cell grows. G2 again cell grow, and M stands for mitosis and S means synthesis or replication of DNA. So D is the correct option. Human body cells in interphase have 46 chromosomes. Yes. Human cells, this is the characteristic of the human cells that they have chromosome number 46. So, already told you that during the replication of DNA in interphase, what happens? The chromosome number is increased. That is, they are replicated. So we have to find out which one statement is correct. 46, as there is one telomere at the end of the 6th chromosome. When the cells will replicate, so according to the replication of DNA, the number will not be 46. So their number will be increased. Now I will move towards the second. 92, as there is one telomer at each end of the 46 chromosomes. Yes, it happens, but as the cell replicate, so it will not. As there is one telomere at the end 42, there are two telomers. So I would like to discuss according to 184, as there is one telomere at each end of the 92. Now I will describe this answer. So I will start with the, that is, during the S phase. Already in the question number 22, I have described that the replication of DNA takes place during interphase. So due to which uh, identical copy of the sister chromatids is created. So these two sister chromatids are joined together at the centromere. So when the cell enters in the prophase, it still has 46 chromosomes, but each one is made up of two sister chromatids. Two sister chromatids, this gives a total of 92 chromatids. 46 chromosome replication in interphase and in prophase 92, and each has two telomers. So we will multiply 92 with the 2, it will be equal to 184. So telomere, is a protective cap found at each end of the DNA molecule or chromatid, you can say. Each chromatid is a separate DNA molecule. It has two ends. So therefore, the number of telomeres will be 184. So according to this, D is the correct option. Next question. 24, sometimes hydrogen bonds form temporarily between the bases of two strands of RNA. How many hydrogen bonds form when guanine and uracil each bind to their complementary bases? Students, there are four complementary bases. They are adenine, guanine, cytosine, and thymine. And in DNA and in RNA, adenine, guanine, cytosine, and thymine is replaced by uracil. So adenine always pair with thymine or here uracil. And guanine, it will always pair with cytosine. So adenine and guanine are purines and thymine and cytosine are pyrimidines. So when adenine pairs with thymine or uracil, there will be the formation of two bonds. And when guanine will pairs with the cytosine, there will be the three hydrogen bonds. So in this way, I would like to say that guanine will make three bonds and uracil, and they will make. So it will be B will be the correct answer. As in DNA, it is thymine, but in RNA, T is replaced by, or thymine is replaced by uracil. Question number 25. And question number 25 is, the table shows all the possible triplet codes for some amino acids. So the amino acid sequence is shown. You can see over here that methionine in the table only one DNA triplet code for methionine. That is the methionine, there is only one code. For isoleucine 1, 2, and 3. So there are 3 for isoleucine, and for cytosine 1 and 2. So 2 for, and so in this way, cytosine, the table shows 2 DNA triplet codes for cytosine. So tryptophan, the table shows only one DNA triplet code for methionine 1, isoleucine 3, and tryptophan 1. So determine the total number of, we will determine the total number of DNA sequences. Uh, to find out this, we will multiply the number of possibilities for each amino acid together. So in this way, I would like to say methionine has one possibility, whereas it has 3 possibilities, and you can see it has 3 possibilities, it has 2 possibility, and again total sequences will be 6. So in this way, a tryptophan will also have 1, but you can see this will be counted as 1, and 1 will be tryptophan and methionine, they will be considered as 1. So I will explain this, that there is a possibility 1. One possibility, 1 is methionine and 1 is for tryptophan. 1 into 2 is equal to 2. And then, so 1 into 1. So possibility is, so here I would like to find out the possibilities. We will multiply the possibility with the, let me explain it as 1 into 1 into 1, sorry, into 2 into 3. So it will be equal to 6. How? Cytosine is 2 times, isoleucine is 3 times, tryptophan is 1 time, and methionine is 1 time. So possibility. So in this way, we will multiply the sequence, number of sequence possibilities. So it will be 1 into 1 into 2 into 3. 1 time tryptophan, 1 time methionine, 2 times cytosine, and 3 times isoleucine. So it will be equal to 6. So we will go with the C. We are not going to plus them. They will not be added. They will be multiplied. Question number 26. Question number 26 is definition of transpiration. Transpiration is the loss of water vapors from the leaves, from the aerial parts of the leaves, especially through the stomata. So according to this, you can see the loss of water from leaf surface. Leaf surface is, you can say, the doubtful statement. As you can say, leaf surface can be the upper surface. So water is, most of the water is lost from the lower surface, or you can say through the stomatas. So evaporation A, B cannot be the correct options. C and D, any one of them can be the correct. So now the second comparison of water potential. You know, the leaf, if the water is in higher potential, then it will move from its higher water potential towards the lower water potential. But higher inside air spaces, if the water is higher in air spaces than the leaf than the air outside. But we have reached that these two, A and B, are not the correct option because of this definition. We will go with the C, loss of water vapors from leaves. Lower inside airspace, if the water potential is higher inside, then it will move or it will be lost into the air or it will be evaporate into the air. So according to this, C is not the correct option. And we will go with the D, loss of water vapors from leaves higher inside air spaces in the leaf than the air outside. Water is higher inside, so it will move outside. Because if water potential is higher inside because of the transpiration pull, water potential will be higher inside, due to which the water will be lost into the air. Question number 27, which features of, so D is the correct option. Which features of companion cells and cell xylem vessel elements make them suitable for their function? So first of all, the companion cells. Companion cells, these are closely associated with sieve tube elements in the phloem. So their primary function is to provide metabolic support, which includes actively loading sucrose into the sieve tubes. So this process requires a significant amount of energy. And energy, it is obtained from the mitochondria. So which are, you can say, found into the companion cells. So according to this, transport contents of cell in one direction only. Cellulose walls provide support. Nuclei allow cell division. Numerous mitochondria supply energy. Yes. Xylem vessel and lignified walls provide support. So the function, we are talking about the function of the xylem, not the support. Nuclei allow cell division. Gaps between cells allow rapid absence of cytoplasm allow mass flow. So these cells are responsible for transporting water and minerals from the roots to the rest of the plant. So perform this function, they are adapted for mass flow. So this is made possible by the absence of cytoplasm. These are the hollow tubes. So these are the hollow tubes due to which, and the end to end, they are joined end to end to make a structure without cytoplasm or other cellular spaces inside. So they create a hollow tube and their lignified walls which prevent them from collapsing under the negative pressure of transpiration. Transpiration pull has a great pressure due to forces, cohesion and adhesion. So the lack of cytoplasm eliminates any obstruction to the flow of water. That is the reason the D is the correct. Water can as easily transport by the xylem vessels. So D is the correct option. Next question is, so here you can see a diagram shows some type root cells. Which statement is correct for the pathway shown by the arrows? Arrows show that water is moving through the cell wall, and this is the apoplast pathway in roots where water and dissolved minerals move through the uh, cell walls and intercellular spaces. So from the root hairs through the cortex until they reach the Casparian strip in the endodermis. So this waterproof strip made of suberin forces the water and minerals to switch to the symplast pathway, allowing the plants to control which substance reach the xylem. So in this way, uh, the water moves into the roots. So water will be blocked by a band of suberin. Yes, this is the correct answer. This is the correct answer. Then water will move through the Casparian strip. Water will not move through the Casparian strip. It will move through the because Casparian is a waxy waterproof band which will block the water movement, so forcing it into the cells rather than allowing it to move between them. So they will adopt the another way. Water will pass easily through the root endodermis. This is, you can say, a general statement. It does not describe the specific pathway which has been shown into the diagram. So according to this, it cannot be. The water will pass through plasmodesmata. Water has is been not shown through plasmodesmata. It is water is moving through the cell wall. Not, it will move, but here you can see it can move, but the arrows show that the water moving from the cytoplasm of one cell to the next through the cell wall. So A is the correct option. So the graph shows the diameter of a tree trunk at different times. You can see at J at 0. 0 shows the midnight. Midnight when at that time uh, largest diameter as comparison to other times. So at this time, it is a midnight and K is the lowest diameter. Again, L shows the highest diameter. So it shows the fluctuations in diameter, which can be due to the movement of the water within the tree xylem. So the tissue responsible for transporting water from the roots to the leaves. So during the day, uh, especially around noon, you can say this time, at this time, you can say the diameter is high as water evaporates uh, from the from the leaves, and less transpiration takes place at that time, due to which the diameter is larger. But as you can say, the K point shows a reduction in the diameter. So it shows a reduction in the diameter, but it can be due to the water loss through transpiration. So you can say the translocation is the movement of sugars through the phloem. So not through the translocation. So again, the at L, the diameter is greater. So I will, J shows the expansion of the trunk. I fill the xylem through transpiration. Expansion is not due to the water filling by the xylem during transpiration. Transpiration causes reduction. K shows a reduction in diameter of the trunk as water is lost from the phloem due to the translocation. Water is not lost due to the phloem. Phloem's function is different. Phloem's function is translocation, not the transpiration. So K shows a reduction in diameter due to the water held in tension in the xylem. Yes. L shows the expansion as the phloem tissue act as a sink at night. Again, this is not shows reduction of the water tension, xylem, and not the phloem acting as a sink. So xylem is in phloem, it is not the sink. It can be the it helps in the translocation, not it acts as a sink. So C is the correct option. and its L shows due to the transpiration. Now, last question number 30 is the plane diagram of transverse section of two plant organs are shown. They are not shown to the scale. In leaves, Q on the upper epidermis, there is xylem, and R is phloem. Again, T is xylem, and S is phloem. So I will go, first of all, I will describe their functions. The point R and T, the point Q is xylem and in T is xylem. So function of xylem is the transport of water. Transport of water, whereas the R and S are the phloem. So, you know the phloem in this, S is the phloem in the stem and R is the phloem in the leaf. So the phloem's primary function is to transport sugars from source, transport of sugar from source, where the carbohydrates are formed towards the sink. They can be roots, shoot, or so like roots or fruits in a process called translocation. So this process is driven by a pressure flow mechanism which relies on the active loading of sucrose into the sieve tubes. So this loading process also involves proton pumps. So this process involves proton pumps. So we will go with the C option, which is phloem. Q and T, they are silent. So they don't need these proton pumps because the sucrose has to be loaded in the phloem and sieve tube elements. So the proton pumps are needed by the phloem. So R and S is the correct option. Students, it was all about today's video. Please don't forget to like, share, and subscribe my channel. Thanks for watching.